Good afternoon, learners. Welcome you all again to this session. Today I will be talking on a particular topic, that is Agrobacterium tumefaciens mediated plant transformation. Now this topic, it belongs to BBYET 143, Economic Botany and Biotechnology, Plant Biotechnology, Unit number 14, Section 14.7, Methods of Genetic Engineering. Now there it is mentioned Agrobacterium tumefaciens under this particular topic, which I am going to discuss in a little bit elaborate fashion. Now in some of my earlier sessions, I discussed on how to generate transgenic plants, and I discussed what are the different applications of the genetic engineering technique in plants. But when you are creating a transgenic plant or generating a transgenic plant, the first and the foremost tool or the technology that you use is vector-mediated gene delivery system. And for the vector-mediated gene delivery system, you have to go through this Agrobacterium tumefaciens mediated plant transformation. Now, this Agrobacterium tumefaciens, first of all, we have to learn about the nature of Agrobacterium tumefaciens, and then what is actually there in Agrobacterium tumefaciens that enables it to be utilized in plant transformation. Because till date, there is no other bacteria that has been utilized for the purpose of plant transformation, excepting Agrobacterium tumefaciens or maybe Agrobacterium rhizogenes. So we have to understand what is there in Agrobacterium tumefaciens that makes it the sole candidate or the sole representative to be utilized in creating transgenics or in plant biotechnology. Now, Agrobacterium is regarded as the nature's genetic engineer, because it is just, as you can see in the picture, it is a rod-shaped soil bacteria. It is a bacteria which is present in the soil, or soil bacterium. It's a rod-shaped bacteria, and it is not merely a bacteria, because it has become now a household name in plant genetic engineering or in plant molecular biology. Because as I said, if you are to generate a transgenic plant through vector mediated system, then there is no other option than to use this particular bacterium. So this has become almost a fundamental item in research in plant biotechnology or in plant molecular biology. The story began almost 100 years ago when two plant pathologists, Irving Smith and Charles Townsend, published a paper in Science. It's a very well-known journal, reporting that a bacterium that they named Bacterium tumefaciens caused plant tumors or crown gall. So Agrobacterium tumefaciens is a bacteria that causes crown gall tumorigenesis or crown gall tumor. The next major discoveries on Agrobacterium and crown gall in the 1940s were made at Rockefeller Institute by Armin Braun, whose interests in the system were initially sparked by a surprising observation of a student working under him studying crown gall in Germany. The student found it very difficult to isolate Agrobacterium from the tumor tissue. So the thing is that they identified that this particular bacterium is causing some abnormality in the plant, particularly Agrobacterium tumefaciens. It is causing crown gall tumorigenesis. So people went on searching what is the reason for this, and then what are the unique characteristics of this particular bacterium. Now, vector-mediated gene transfer. So what does it mean? That means gene transfer, or when you generate a transgenic plant, it can be done in two ways. Either you can undergo vector-mediated DNA delivery, or it can be direct gene transfer. Direct gene transfer means when there is no involvement of the vector. That means it can be done through particle bombardment, gene gun. It can be done through electroporation. Okay, so there are several other techniques. But when you are talking of Agrobacterium mediated transformation, you have to understand that it is a vector mediated gene delivery system or vector mediated DNA transfer. So from this it is very clear that Agrobacterium tumefaciens must have some components, must have some inbuilt components, or must have some vector within itself, which actually we make use of in plant transformation. So we will have to study in details about that particular vector which is present endogenously in Agrobacterium tumefaciens. And that plasmid or that vector is known as the TI plasmid, because we already know that plasmids are a kind of vectors. So these Agrobacterium tumefaciens, they have this TI plasmid or the tumor inducing plasmid, and that is why the tumors are being generated in the plants. Now in this context, I would like to mention that in addition to Agrobacterium tumefaciens, you have another one species of Agrobacterium which is called Agrobacterium rhizogenes. And that Agrobacterium rhizogenes, it causes root mat disease, or it causes hairy root diseases, where there is a huge proliferation of the root growth and there is abnormal root formation. So we will not go into details of Agrobacterium rhizogenes today. We will be focusing our attention mostly on Agrobacterium tumefaciens. Now, Agrobacterium tumefaciens is a soil-borne gram-negative bacterium causing crown gall disease. It's a rod-shaped motile bacterium which is found in the rhizosphere. So you all know what is meant by rhizosphere. That is the region of the root which is in immediate vicinity with the soil. And there this particular bacterium is housed, or it is a resident of that particular region. But what happens is, whenever there is an wound, whenever a plant is wounded or whenever there is a damage, then you find that the crown gall formation is formed at the damaged site. Now it is not necessary that the damage has to be near the soil, near the level of the soil. It can be quite high. Like in a lofty tree, there can be a damage at quite a substantial height from the soil surface, and there the crown gall is formed. And as I said, that crown gall is caused due to Agrobacterium tumefaciens. Then you have to understand that there is some sort of movement from the soil particle to the top of the tree, and that movement, it occurs due to chemotaxis. Why chemotaxis? Because from the wounded site or from the damaged site, certain kinds of molecules are being secreted, like different kinds of sugars or certain phenolic molecules. These are being released from the damaged plant cells. And in response to those chemicals, Agrobacterium, it shows chemotactic movement, and it moves from the soil to the damaged site. And thereafter it actually colonizes at the damaged site. So these are the three species, as you can see, that is Agrobacterium tumefaciens crown gall disease, Agrobacterium rhizogenes hairy root disease, Agrobacterium radiobacter, it's an avirulent strain. Now today, as I said, I am focusing my discussion on Agrobacterium tumefaciens. Now before I begin actually the Agrobacterium tumefaciens biology, we categorize Agrobacterium tumefaciens strains into two groups, that is biotype 1 and biotype 2. Biotype 1, it includes certain strains like ACH5, B6, okay, T37 and so on. And these strains, they normally grow up to 37 degrees centigrade. But normally they grow at 28 degrees centigrade. Okay, even if you raise the temperature, they might tolerate up to 37 but not beyond that. But it is preferable that you culture them. When you are culturing them in the laboratory, then you culture them preferably at 28 degrees centigrade. And what are the media that which you can use here? You can use the Luria-Bartoni media, which you use in case of Escherichia coli or E. coli. You can use nutrient broth, or you can use tryptone yeast extract. Now, in case of biotype 2, they have a different requirement. That is, they grow on erythritol as a carbon source. And such strains like you can see NC, PPB, 1855, A4, etc., they normally proliferate poorly at temperature higher than 30 degrees centigrade. So here the temperature range is much narrower, and hence it is preferable that it is recommended that you should grow them at 28 degree or 29 degrees centigrade. So minor fluctuations in temperature might arrest the growth of these kind of strains. So you have to be very careful regarding the maintenance of proper temperature. And they do not grow on classic E. coli media. Instead, they can be cultured on tryptone yeast extract plus calcium. So they need certain supplemented media and not the normal media which you use in case of E. coli. So these are the two strains, and you can use either of the two strains for biotype 1 or biotype 2 in case of plant transformation. Now, as I said that Agrobacterium tumefaciens, it has the tumor inducing principle, or it has the TI plasmid. So we have to now understand very clearly what is the structure of this TI plasmid and how do you clone a particular gene in the TI plasmid, what are the different ways by which you can undergo the cloning and the transformation process, because we are utilizing this TI plasmid for plant transformation. If it is Agrobacterium rhizogenes, then they have the RI plasmid, or the as I said that that actually induces the root hair formation, hairy root disease as we say. So those rhizogenes, they have the RI plasmids. Again, I am not talking of the RI plasmid. I will be mostly talking on the TI plasmid. Now, some of the points which need to be remembered when you are undergoing Agrobacterium tumefaciens transformation in your laboratory. Number 1, continued presence of viable bacteria is not required for tumor maintenance. Once you infect the particular plant tissue, it can be callus, it can be leaf discs. Once you infect the particular tissue with Agrobacterium tumefaciens, okay, and if the Agrobacterium colonizes, okay, then it's not necessary that the Agrobacterium tumefaciens has to be present continuously. Okay, once the infection is done for a certain period of time, then you need to remove the Agrobacterium tumefaciens. Otherwise, you will not be able to regenerate the plant. You will not be able to regenerate a plant from the transformed cells. So initially, for a certain period of time, I will talk on that. For a certain period of time, you need the co-cultivation. Co-cultivation means where you are growing the Agrobacterium together with the plant tissue or the explant. And after that period is over, you have to kill the Agrobacterium tumefaciens cells and you allow the explant, transformed explant, to be regenerated. So this is number 1. Number 2, bacteria do not penetrate into plant cells that are converted to tumor cells. Number 3, rather bacteria, it penetrate into the intercellular spaces and into the injured cells and attach themselves only to the wall of the healthy plant cells. Now, what enables Agrobacterium tumefaciens to attach to the walls, we will be coming to that. Of course, it has certain genes. Okay, attachment which helps in the attachment, it has certain genes, and those genes enable the Agrobacterium tumefaciens to attach to the plant cell wall. Now take a look very carefully at this particular figure. Now this is actually the TI plasmid. So the TI plasmid has different components. As you can see, it has a right DNA border, right tDNA border. And what is tDNA? tDNA means this particular region, that is, which is present within the border sequences. So these are called the border sequences. One is the right tDNA border and another is the left tDNA border. And within this region, this particular region between these two borders are known as the tDNA region or transfer DNA region, meaning that only this portion of the DNA will be transferred into the plant cells. The rest of the portions will not be transferred. But you have to remember that the other portions are also important so far as the transformation process is concerned. It's not that these are not important, but only the tDNA region will be transferred. So the right tDNA border and the left tDNA border, within this region, within the tDNA region, you have certain hormone regulating genes, or you have certain genes which regulate hormone production like auxin and cytokinin. So auxin production and cytokinin production, and the genes which are responsible for this production are also present within the tDNA region. In addition, you have a certain component called opines. I will come to that. the opines are actually synthesized not by the bacteria, but the opines are synthesized by the infected plant cells, which enable the bacteria to grow. That means these opines, they serve as carbon and nitrogen sources for the Agrobacterium tumefaciens cells to grow. And these opines are not synthesized by Agrobacterium, rather they are synthesized by the infected plant cells for the bacterium. So the opine synthesis genes are present actually in the tDNA region, and you can see an interesting thing, that oncogenes. So by the term oncogenes, we normally mean the genes which are responsible for cancer formation. But you should be surprising that you should be surprised to see that why the oncogenes is there in this particular TI plasmid. Because if there is an abnormality in the auxin and cytokinin production in the plant system, then there will be abnormality. And that is why these genes are also sometimes referred to as the oncogenes. Because if these genes, they get mutated, the normal genes which regulate auxin and cytokinin production, if they get mutated, or if they become non-functional, or if they become function in an improper fashion, then that might lead to oncogenicity of the particular plant, because it might lead to abnormality. And you can imagine crown gall to be a kind of cancer. You can imagine the hairy root to be a kind of cancer in case of plants. So if the question is that do the plants have oncogenes, the answer is yes. Because if there is any kind of change or mutation or any kind of abnormality in the genes which are responsible for auxin and cytokinin production, there will be a cancer formation in plant as well. So these are termed as the oncogenes. So these oncogenes are present within this particular tDNA region. And outside the tDNA region, you can see the conjugative transfer process, because the Agrobacterium will actually get transferred into the plant cells. So there should be some kind of transfer, this transfer process regulating component, and that is known as the TRA locus. Okay, that is present here, that is responsible for the conjugative transfer. Then there is a component called opine catabolism. That means opine synthesis is within the tDNA region. Opine catabolism genes are present here, that is outside the tDNA region. The origin of replication, which allows the vector to multiply within the host cell, and a very, very important component which we will focus in detail, that is the virulence region. That means it has almost 8 virulence genes, or the VIR genes. And these VIR genes, they do not, they act from outside. That is, they allow this tDNA region to be entered or to gain entry into the plant cells. But themselves, they do not get entry into the plant cell. So with this picture in our background, we will try to focus on each of these components in a little bit further detail. How they are being regulated, and what function do they precisely regulate during the transformation process. So number one, only a small part of the tDNA, only a small part of the tumor inducing plasmid is being transferred. That is, only the tDNA region will be transferred. Now this tDNA region, it contains genes for hormone synthesis, that is auxin and cytokinin, and the biosynthesis of novel plant metabolites called opines. Now opines can be again of different kind, like agropine, nopaline, octapine, and so on. But each TI plasmid will have only one opine synthetic gene or opine catabolizing gene. Because so accordingly you can name the TI plasmid as octopine plasmid, nopaline plasmid, and so on. Production of auxin and cytokinin causes plant cells to proliferate and hence form crown galls. As I told you, that if there is an imbalance between the auxin and cytokinin, there will be an abnormality. If the auxin concentration becomes too high, then obviously there will be profuse root formation, and if the cytokinin level is too high as compared to auxin, then there will be the crown gall formation. Okay, so both are kind of cancers, or both are kind of oncogenic effect, and therefore a proper balance between cytokinin and auxin is actually necessary, and that is why they are known as the oncogenes. Now, as I said, the proliferating plant cell, the infected plant cell, they are also secreting certain amino acid derivatives called opines. Mind that these opines are not utilized by the plant, but they are utilized by the bacteria. The plant actually synthesizes them. But again, a healthy plant will not synthesize opine. Only an infected plant cell will synthesize opine, and they will be utilized by the bacterium as carbon and energy sources. Now octopine, like octopine, it has carboxyethyl derivatives of arginine. Nopaline, it has dicarboxypropyl derivatives of arginine. Agropine, it has bicyclic sugar derivative of glutamic acid. So the most of these octopines, they are some amino acid derivatives, but there are slight differences in their structures or the components. So accordingly, you have the synthesis genes which are present in the tDNA region, that is octopine synthase, nopaline synthase, and so on. And TI plasmids, the nomenclature of TI plasmids are done on the basis of these opines, that is octopine TI plasmid, nopaline TI plasmid, and so on. A bacterial strain, it produces only one type of opine, and it also catabolizes only that opine. For example, if it is an octopine TI plasmid, then there is no question that it will synthesize nopaline, or there is no question that it will utilize nopaline. It will only synthesize and it will only utilize octopine. So it's very much specific. Opine catabolism is regulated by certain other loci, like it can be OCC, it can be NOC, and so on. That is catabolic genes. So the synthesis genes and the catabolic genes, the production and the degradation, for each of these there are separate genes. Now, how does a transformed cell differ from a non-transformed tissue? When you infect a cell with Agrobacterium, then their certain properties get changed. What are those change in properties? It is able to grow autonomously on synthetic media. The symptoms or the phenotypes or the changes are similar to that what happens in case of cancer in human cells, because a cancer cell, it also loses all kind of control. That is, it grows autonomously, or it grows in a very proliferating fashion. Okay, even if you don't apply the important components in the growth media, okay, it is able to proliferate. Here also, it is a phytohormone independent growth. You don't have to provide phytohormones. Okay, you don't have to provide auxin, cytokinin and all. It will grow on like that only. Okay, so this is number one, unlike a normal tissue or a healthy tissue. Number two, plant cells contain one or more of the unusual amino acid derivatives called opines. This is another difference. I told you, healthy cells, they do not have the opines. Only the infected cells, they have the opines, because that is being utilized by the bacteria. So these are the two basic differences. Now, what is meant by genetic colonization? Genetic colonization means, so one thing is very clear, that if I ask, what is the reason that only Agrobacterium tumefaciens is capable of transforming plants? No other bacteria till date have been found to be involved in this plant transformation process. Then there should be something in Agrobacterium tumefaciens which enables it to achieve this quality. And the reason for that is the opine, because opines actually distinguish Agrobacterium from other bacteria in the rhizosphere, because till date, Agrobacterium is the only bacteria which is capable of utilizing opines. No other bacteria can utilize opines. So this actually provides Agrobacterium a competitive advantage or a selective advantage over other bacteria. So other bacteria, even if they try to infect the plant, they will not be able to infect the plant in the same fashion, and they cannot be utilized for plant transformation. So this is number one. And number two, this interkingdom gene transfer. That is why it is interkingdom, because from bacteria, the gene or the vector is being transformed into another system, which is a plant system. So this is something, a beauty, that from a bacteria, you are transferring something to a plant. Normally in conjugation process, we have seen that from a bacteria to another bacteria. But here, from a bacteria to a plant system, a component is being transferred. So this is really a beauty, and this interkingdom gene transfer is termed as genetic colonization. So the term genetic colonization, it can have two connotations. One is the issue of the opine utilization, and number two is the interkingdom gene transfer. Now, in addition, there are certain chromosomal genes in Agrobacterium, like CHV genes, And as I told, that Agrobacterium needs to attach itself to the wounded site. Whenever there is a wound, the Agrobacterium will undergo chemotactic movement, and therefore it will colonize there and it will attach to the cell, okay, to the wounded site. And for that there are certain genes which are called as the chromosomal genes, like CHVA. As you can see, it's a constitutively expressed non-regulated genes. It encodes an inner membrane protein essential for transport of cyclic beta-1,2 glucan from cytoplasm to periplasm. CHVB, it encodes an inner membrane protein involved in synthesis of beta-1,2 glucan of attachment polysaccharides. CHVD and CHVE, they help in the optimal expression of Wnt genes. I have not come to Wnt genes yet, but you have seen that there is a virulence cassette outside the tDNA region, and that virulence cassette, or the stack of virulence genes, there are 8 virulence genes which are working in the form of an operon, that is Vir A to Vir H, Vir A, B, C, D, E, F, G, H. There are 8 such genes which are working in a operon fashion, operon system, and that Wnt genes are also regulated by this CHVD and CHVE. In addition, there is an exolocus gene which helps in the biosynthesis of attachment polysaccharides. So in a nutshell, what I mean to say is that this attachment of the Agrobacterium to the infected cell or to the wounded site is mediated by a set of or a cluster of chromosomal genes. And when the bacteria will attach to the cell wall, we all know that the cell wall has different polysaccharides. It has the cellulose, okay, it has the hemicellulose, it has the pectin, of which the cellulose is the major component, and cellulosic polysaccharides. So the Agrobacterium, ultimately it has to attach to those cellulosic polysaccharides, and for that it has this entire set of chromosomal genes. Now we will go into each and every component of the TI plasmid in further details. Now this TI plasmid is a huge plasmid. It is a 200 kilobase pair length plasmid, and it exists as independent replicating genetic units because it has its own origin of replication. So it can autonomously replicate in any system, okay. And Nopaline TI plasmid, it has a single 23 kilobase pair tDNA region, but Octopine type TI plasmid, it has 2 adjoining regions of 13 kilobase pair left hand piece and 8 kilobase pair right hand piece, of which only the TL region is oncogenic. That means you have to understand that the tDNA region is interrupted. It's not continuous. In case of Nopaline type, it is continuous, but in case of Octopine, it is interrupted, and the auxin and cytokinin regulating genes are present in the TL region, and that is why we call the TL region to be the oncogenic, because by this time probably you have got the idea what is meant by oncogenicity in case of plant system. Nopaline tDNA region, it has 13 ORFs, while Octopine tDNA, it has 8 ORFs in TL and 16 ORF. ORF mean the open reading frames. For Nopaline plasmid, TL and T are transferred together, but in case of Octopine type, they are transferred independently. And what are these oncogenic regions? Oncogenic loci, that is rooty locus or tumor morphology root, which is designated as TMR, and results in tumors developing abnormal roots, and shooty locus or tumor morphology shoot or TMS, which results in tumors with abnormal shoot tissue. So if there is abnormal root formation, you have to understand that it's a hairy root kind of system, or we also call it root mat disease. Or if it is an abnormal shoot tissue, then you have to understand that there is a crown gall formation. Now, mutation of a further morphogenetic locus, TML, it produces larger than normal tumors. So in case of auxin biosynthesis, what are the genes? AUXA and AUXB, which are also otherwise known as TMS1 or TMS2. And in case of cytokinin production, you have the TMR. It encodes isopentenyl transferase that catalyze isopentyl adenosine 5' monophosphate or cytokinin. So there should be a proper regulation of these genes, because if there is any kind of perturbation in the regulation of these genes, it might lead to an abnormality. Either it might go towards the hairy root formation or it might go towards the crown gall formation, and that is why these genes are regarded as oncogenes. Another gene, it encodes the enzyme dimethyl allyl pyrophosphate AMP transferase or the DMA transferase. It helps in the covalent linkage of dimethyl allyl pyrophosphate to the N6 nitrogen 6 of adenosine monophosphate, yielding isopentenyl adenosine 5' monophosphate, which is the first cytokinin in the metabolic pathway of this phytohormone. So this is another cytokinin regulating gene which is also there within the tDNA region. Now we are coming to another issue. So that we have completed discussion on the oncogenes. Now we will come to the next component. If you remember that picture of the TI plasmid, you have seen that the tDNA region is actually the two borders of the tDNA region. You have some border sequences, that is the right tDNA border and the left tDNA border, and we showed that only the tDNA region which is present between the border sequence is transferred. Now how is this possible? And what are actually there in these border sequences? I mean, what these border sequences are actually made up of, and how it is actually occurring. That means only the tDNA region is occurring. So you have to understand that probably there is some sort of mechanism by which there is a cutting, because unless there is a nicking or unless there is a cutting mechanism somewhere, then how can you excise this particular portion so that it can be transferred, and it has to separate from the rest of the TI plasmid. So we will now be discussing about that and what are the different proteins that are actually helping in this cutting or nicking. Now mind that this cutting or nicking will occur only at the right tDNA border and not on the left tDNA border. So it is very clear that the transfer process will be initiated or will be started from the right tDNA region, and it will continue till the transfer process reaches the left tDNA region. All the components within that region will be transferred. So whatever gene you need to clone or whatever gene you need to transfer to the plant to make a transgenic, to create a transgenic, has to be housed within the tDNA region and not outside, because otherwise those will not be transferred. So the site of endonuclease action during tDNA transfer is the 24 base pair highly conserved directly repeated tDNA sequences that flanks and delimits the tDNA. So tDNA is separated from the rest of the TI plasmid by the presence of these border sequences. And it is the right tDNA border is the site of the endonuclease action. And as I said, any sequence between this will be transferred. Okay. The TL region of octopine PTI, genes for auxin and cytokinin biosynthesis. TR region, it has genes for agropine and manopine synthesis. Okay. And then the region outside the tDNA, we have seen that there is a 40 kilobase pair virulence region or the VIR genes, and there are 8 operons, as I said, VIR A to VIR H, and they are co-regulated, and therefore we call it an operon system. They are co-transcribed. Now these virulence genes, they are not transferred to the plant. They don't gain entry into the plant. Then you must be wondering what is their utility. If they are not transferred in the plant system, then what is their utility? Why they are required? They are required for the transfer process. They help in the transfer process, but itself they don't get transferred into the system. Now we have to look at how they are helping in this transfer process. Like VIR A, VIR B, VIR D, VIR G, these are essential for virulence, and the rest of the VIR genes, they play an accessory role. VIR A and VIR G, they make up a two-component regulatory system important for transcriptional activation of the remaining VIR genes and to some extent their own increased activation. VIR regions are located outside the tDNA region. Now, whenever there is an wounding, okay, so this wounding actually it induces the VIR genes, and there are certain chemicals which are released from the wounded site, which are known as the acetylseringon or beta hydroxyacetylseringon or alpha hydroxyacetylseringon. Now, why the Agrobacterium, it actually gets mobilized, or it actually undergoes chemotactic movement? Because in response to these signaling molecules, so from the soil it will go to the wounded site, because wounding, it induces the virulence genes, and two of the most active substances are identified, and the VIR-A is a product of the periplasmic membrane protein that senses these phenolic components. So they are phenolic sensors, and in response to that, Agrobacterium actually mobilizes, it moves, or it undergoes the chemotactic movement. VIR-A is a phenolic sensor autokinase and is a receptor for acetylseringon. So there is an autophosphorylation of VIR-A, and thereafter, after it undergoes this phosphorylation mechanism, it transfers the phosphate group to the VIR-G, and therefore VIR-G gets activated. So VIR-A is an autokinase, there is an autophosphorylation, and thereafter it transfers the phosphate group to VIR-G, VIR-G gets activated, and it is a transcription factor, which means that several genes are under the control of VIR-G. That is, when VIR-G gets activated, there are several downstream VIR genes. Which are those downstream VIR genes? That means other than VIR-A and VIR-G, all the downstream VIR genes, they are directly regulated by VIR-G, and therefore we call them as a VIR box. And a series of VIR genes, they are being transcribed one after the other to regulate the DNA transfer process, or the tDNA transfer process. So VIR-A is a phenolic sensor and an autokinase, and VIR-G is a transcription factor which gets activated by phosphorylation. Now, VIR-B1 to B11, these are membrane proteins which help in the transfer process by acting as the transfer apparatus for the tDNA transfer process, and they form a channel for the tDNA transport. VIR-B11, it has ATPase activity, because for this transfer process, successful transfer process, it needs energy, and that energy, it comes from ATP hydrolysis, and VIR-B11 is it has an ATPase activity. VIR-C is a helicase and an overdrive binding protein. Overdrive sequences are present just outside the right borders, and these overdrive sequences are recognized by VIR-C, which is a helicase, and that is why probably the reason that the transfer process starts from the right border and not the left border. It is because of the presence of these overdrive sequences which are present near the right borders, and which are being recognized by VIR-C, which has a helicase activity. And helicase means that there is an unwinding of DNA. So from the double-stranded DNA, it has to get converted to the single-stranded form before the transfer process is being initiated. And that occurs exclusively at the right tDNA region. VIR-D1, it has a topoisomerase activity which binds to the right border of the tDNA required for tDNA processing. VIR-D2 has an endonuclease activity. This is very important because, as I said, that there is a nicking, there is a cutting at the right DNA border, and it directs tDNA through VIR-B, VIR-D4 transfer apparatus. Like in case of conjugation, you have a conjugation bridge. Okay, here you don't call it a conjugation bridge, but here also you need some kind of transfer apparatus. You need some kind of channel through which the DNA has to transfer, and that transfer apparatus is made of VIR-B or VIR-D4. Now, when you undergo Agrobacterium transformation, you have to understand that the DNA will enter directly into the nucleus. It does not go into mitochondria and chloroplast. Why? Because all these VIR genes, they have certain nuclear localization signals. And when the DNA is being transferred from the Agrobacterium to the plant cell, there are certain VIR genes which will also bind to them. Okay, and they will ensure that the DNA is being mobilized, or the DNA gains entry into the nucleus and not on any other subcellular organelles like mitochondria or chloroplast, because they also have the DNA in multiple copies. But transformation through Agrobacterium is a nuclear transformation, meaning that the DNA will enter only into the nuclear genome, integrated into the nuclear genome, and for that these VIR genes, some of the VIR genes are responsible, like VIR-D2. So VIR-D2 has a number of activities to play. Karyofurin alpha, cyclophilins, PP2Cs are the interacting proteins involved in the transfer of proteins through the nuclear pore complex. So VD1 and VD2, they provide an endonuclease system which initiates the nicking process. They produce single-stranded nicks at specific sites in DNA. Nicking takes place between the third and fourth base of the bottom strand of each 25 base pair repeat. A nick at the right border 25 base pair repeat provides a priming end, and synthesis of new DNA strand begins from this nick in 5' to 3' direction. So the transfer has to occur in a single-stranded form, and after the transfer process is being done, then there is a reformation of the DNA synthesis. That means again this nick site will act as the priming site for the synthesis of the new DNA strand. So the DNA transfer process, it occurs in a single-stranded form. VD2 becomes covalently attached to the 5' end of the displaced single-stranded tDNA strand. Presence of VD2 makes the 5' end of the T strand less vulnerable to an attack by exonuclease. So it is an endonuclease attack at the right tDNA region. Outside the tDNA, immediately adjacent to the right border, as I already mentioned, that there are some short sequence called overdrive sequences, and VC1 and VC2, they actually recognize these overdrive sequences, and therefore, as I said, that the transfer process, it starts from the right border and not the left border. tDNA VD2 complex cross the plasma membrane to the cytoplasm. The tDNA strand is covered with VE2. What is the function of this VE2? VE2 actually protects the tDNA from nucleases, because see, this tDNA region is entering into a foreign system, that is plant, and the plant will try to actually degrade any kind of foreign material. Any system, it tries to degrade anything which is recognized as foreign. So there will be a tendency for the plant cell to degrade the foreign DNA, but that is not done. That is not possible. Why? Because VE2 is there. VE2 actually protects the tDNA so that they are not degraded in the plant system, and it facilitates the nuclear localization process. VE2 is also a 69 kilodalton single-stranded binding protein, and 600 copies of such VE2 binds to the single-stranded tDNA region, and even this VE2, it has 2 nuclear localization signals. And in addition, VE2 has also another interacting protein, that is VIP1. This VIP1 also interacts with VE2, and together they actually enable the entire transfer process. So see, the tDNA is not transferred alone. There are several additional VIR proteins which bind to the tDNA region, okay, and enables it to get transferred through the transfer apparatus via the nuclear pore complex, via the nuclear membrane, and then enter into the nucleus to be integrated or to be incorporated in the nuclear genome. So it's a beautiful kind of mechanism. It's a well orchestrated mechanism which is regulated by a series of VIR genes. Okay, and VIP1 and VE3 facilitate interaction between VE2 and karyofurin alpha, which transports VE2 into the nucleus. VE2 gives correct conformation to the tDNA strand. Another VIR gene, that is VIRF, it is a bacterial F-box protein, and it helps in VIP1 and VE2 degradation by proteasomal machinery. So this is almost towards the end where VIRF actually works. Once the tDNA region has entered into the nucleus and is going to be integrated into the nuclear genome, then there is no requirement for these VIR proteins. There is no further requirement of these VIR proteins. So these VIR proteins need to be degraded, and therefore there is this VIR gene, that is, or VIR protein, that is VIRF, which actually targets these VIR proteins to undergo proteasomal degradation process, and these proteins are finally degraded. So the entire process or the entire integration of the tDNA region, it occurs by illegitimate recombination, unlike homologous recombination. So you can see that what are the different functions of VIR genes, And in addition, it also has VH, which consists of 2 genes, which has a role in the detoxification of certain plant compounds that might adversely affect the growth of Agrobacterium. So this is actually the entire Agrobacterium biology, where you have learnt about the oncogenes, you have learnt about the opines, you have learnt about the roles of the different virulence genes which act from outside. They do not gain entry into the system, but even if they gain entry into the system, they are ultimately degraded. Okay, their main task is to enable the tDNA region to get transferred and to get integrated. That is the main purpose of weed genes. You have learnt about the utility of the border sequences. What are there actually in these border sequences, and who actually recognizes these border sequences, how the nicking process is occurring, and then how the transfer process actually stops at the left tDNA border sequences. So with this picture in mind, now if you are given in your laboratory to undergo a plant transformation process using Agrobacterium tumefaciens culture, then how will you go, or what are the steps? Okay, so before going into the laboratory part, if we just try to summarize what are the steps. Number 1, there is a signal recognition. Signal recognition by the Agrobacterium, that is from the wounded site there are certain chemicals which are released which will be recognized by Agrobacterium. Then it will attach to the plant cells. We have already seen attachment via polysaccharides. Cellulosic fibers are there to which the Agrobacterium cells will attach. Then there will be the induction of the virulence genes in response to the chemical molecules secreted like acetylseringon or alpha hydroxyacetylseringon, which is released from the wounded site. There will be the weed gene triggering. Then there will be the T strand production. Then the left border and right border sequences will be recognized. Then there will be a cutting or single-stranded nicks at the VD2 locus, and then the VD2 will become covalently attached to the 5' end of the displaced single-stranded tDNA strand, and several other V proteins will actually bind. Several V proteins will form the transfer apparatus and so on. Finally, the tDNA transfer process will occur, and the tDNA VD2 complex is exported from the bacterial cell by T pilus, like we call as the sex pilus in case of conjugation. Here we call it as a T pilus, composed of proteins encoded by VB operon, and VD4E2 and VREF are also exported from the bacterial cell. So those are actually the different steps. In a nutshell, the steps for the Agrobacterium transformation. Now, what is the obvious advantage of Agrobacterium transformation? They are capable of infecting. You are capable of you can infect intact cells, tissues, and organs with rapid regeneration. Then they induce less rearrangement of transgene. Like in case of direct gene transfer, particle bombardment, there occurs rearrangement. Here there is less rearrangement. Lower transgene copy number and stable integration and expression. So these are certain advantages for which people normally choose Agrobacterium tumefaciens mediated transformation. What are the important factors that need to be considered? Like the plant tissue that you are transforming, that has to be competent. It should be a healthy plant tissue. It should not have abnormality. It should not be diseased. Number 2, the vectors should be derivatives of Ti with only left border and right border, and it should have selectable markers because you have to undergo antibiotic selection. It should have the origin of replication like any other vector. But Agrobacterium tumefaciens also has certain shortcomings. It has a limited host range because for a long time people actually succeeded in Agrobacterium tumefaciens transformation only with the dicot plant, not in monocot. Why? Because the dicot plant, they secrete a larger amount of that is acetylseringon or alpha hydroxyacetylseringon. But for monocots, the concentration or the level of these sensors are very less, and therefore it is very difficult to undergo rice transformation or any monocot transformation using Agrobacterium. But the dicots have they secrete this kind of molecules or metabolites to a higher extent. So for monocots, you have to supply this kind of sensors into the growth media from outside if you are to go for successful transformation. Sometimes there are certain cells that are able to regenerate but are difficult to transform. So in this case, you can actually regenerate them, okay, but you cannot transform them easily, but they are easy for regeneration. The reverse also is there, okay, where the transformation process is easy, but the regeneration of the explant, okay, the transformed explant is difficult. So those kind of things need to be considered. Then, as I said, in case of monocots, there are certain secondary metabolites like DIMBOA which are released, which actually inhibits the growth of Agrobacterium. And lack of wound response from monocot tissue, because dicots, they secrete a higher amount of the phenolic sensors, as I said. Certain strains of Agrobacterium which you use, like LBA 4404, you can use it for, this is a very routine, it's a strain which is routinely used for transformation. And in case of monocots, as I said, that they need additional super virulent strains for transformation, like EHA 101 or EHA 105, because they have extra copies of virulence genes. Okay, so in case of monocot, it's better to go for these strains, whereas in case of dicot, you can use anyone, like in tobacco transformation or any dicot species, if you, or Arabidopsis, or in case of, let's say, tobacco particularly, you can use LBA 4404. In case of rice or any other monocots, you can use this kind of EHA 101 or 105. Now, when you are doing this transformation in a lab, what you do is, like, you prepare small leaf discs, which are, you surface sterilize a leaf and you prepare small leaf discs. You infect the leaf discs with the Agrobacterium culture, okay, and grow them to certain OD. And then if they release acetylsyringon, it's fine. Otherwise, you apply in the media the acetylsyringon. The explants are blood dried and transferred to the culture media. Then there will be multiplication. And then after co-cultivation, that is growing the Agrobacterium to the infected tissue, what you do is, like 48 hours growth, you apply a bacteriostatic agent, that is cefotaxime or carbenicillin, and then the transformed calli are, then the bacteria are killed, and then the transformed calli are, they are placed on the antibiotic media for regeneration of the plant tissue, and then when the plant is regenerated, they are hardened and they are transferred to the greenhouse condition, and then you can undergo all the molecular analysis. I am not talking of those molecular analysis because those have already been discussed in one of my earlier sessions. How do you actually check a transgenic plant? That is through southern hybridization, through RT-PCR or northern blot, you check the gene expression, and through western blot, you check the protein expression. So I am not repeating the same thing again and again. Okay, so with this picture in our background, I think today what we discussed is the main Agrobacterium tumefaciens mediated transformation. What are the different components? How will you undergo an Agrobacterium transformation in the laboratory condition? In addition, I would like to say that there are certain other modified techniques like floral dip method or vacuum infiltration method. These are also upcoming techniques, or these are also being simultaneously practiced with Agrobacterium. Particularly the floral dip method, what happens is, supposing you have an inflorescence and you dip the inflorescence in the Agrobacterium culture. So here you don't need to cut or to cut a plant tissue to make an explant. So you can directly dip the flowers or the inflorescence part in the Agrobacterium culture, okay, and you can use vacuum infiltration method, or you can use any surfactant, sucrose and surfactant, for this transformation process, and then you generate the seeds. Okay, after transformation, you generate the seeds, you harvest the seeds and grow them in the selection media, that is in the antibiotic selection media, and you regenerate the transformed plants. So here you don't need any kind of dissection, or you don't need any kind of cutting. So those kind of techniques, advanced techniques, are coming up with the Agrobacterium tumefaciens, and many laboratories are undergoing those kind of techniques. But for you students or learners, you should focus mostly on the Agrobacterium biology, Agrobacterium transformation, and the main components, that is the TI plasmid of Agrobacterium tumefaciens. So with this, I thank you all for very patient hearing, and in case of any inquiry and in case of any problem, okay, you are free to contact us, you are free to write us, you are free to email us, and I wish you all a very, very happy learning. Please go through this Agrobacterium biology very carefully. Thank you.